We will start in a minute or two. Hello, everyone. Welcome to Cell Culture Technologies Week 2 live session. Uh, am I audible? Someone can answer.
Yes, ma'am.
Okay. Is my presentation visible?
Yes, ma'am.
Yes, ma'am.
Okay. So let's start. So, in week 2 of Cell Culture and Technology, you would have watched the videos. It included biology of cell culture, layouts and designs of cell culture facility, precautions during designing the lab layout 1, 2, and 3. So I just wanted to give a brief about do's and don'ts in cell culture and what all do's are like mandatory in all types of cell culture and the don'ts which are of utmost importance. So let's start with that. At any time, if you are unable to see my presentation or my audio is not audible to you, please let me know via mic because I will not be checking the chat box. Okay, so let's start. So the basic do's of cell culture. First is use personal protective equipment, that is laboratory gown, your lab coats, gloves and eye protection at all times. So laboratory gloves and laboratory coat are very important. Eye protection is also encouraged if you have anything that is harmful to your eyes, like anything that is volatile or anything that is it has the capacity of fuming and may cause eye irritation or if even in infectious biology labs too. And in addition, thermally insulated gloves, full face visor, splash proof apron should be worn when handling liquid nitrogen. So in the previous session, we spoke about freezing the cells in liquid nitrogen. So when you do that, the liquid nitrogen has the tendency to vaporize very quickly. So it releases a lot of nitrogen as soon as you open it. So at that time, it is very important to have all these things that are given here, that is insulated thermal gloves. Otherwise, it will cause skin irritation, skin erosion, because whatever the liquid nitrogen touches, it freezes it immediately. So even your skin will get frozen and you will get frostbite sort of condition. And then full face visor, obviously to protect your face. And then splash proof apron. So your apron also should be made of a material that if any harmful chemicals or substances like liquid nitrogen or HCl or hypochlorite falls or splashes accidentally on your apron, the apron should be able to tolerate that and it should not seep in through your clothes within your apron. Next is always use disposable caps to cover your hair. So this is also very important because not only because you might shed hair in the sterile environment, but also many of us have dandruff or any kind of condition. So dandruff, as you all know, is like fungal, so this can spread in the lab if uh, it can become a sort of an aerosol, or when you accidentally bump your head or some sort of disturbance happens and some flakes of dandruff or anything, sweat from your scalp falls in the sterile environment, it will get contaminated. So for men, it is preferred to have a properly groomed hair, maintain self hygiene, and for women also maintain self hygiene. And when you have long hair, tie it up using a hair tie, rubber band, hair band, anything. And men who have long hair also shall follow the same. And on top of that, you can wear a cap to cover your hair. Next, wear dedicated PPE for tissue culture facility and keep separate from PPE worn in general laboratory environment. The use of different colored gowns in laboratory coats makes this easier to enforce. So this depends on the lab practice. In most of the labs, which are basic level labs, might not use PPE, and we can wear the splash proof aprons, gloves, eye protection wear, hair cap, etc. That should be enough. But if your lab is highly susceptible to contamination, or if the organism which you're working itself is highly infectious, then like a virus, HIV, Ebola sorts of virus which can cause a breakout of disease, and it can spread in the population and then become a epidemic pandemic, it's always mandatory to wear a PPE gowns. Next, keep all work surfaces free of clutter. So this is a very basic and very important thing. Let it be your cell culture room, let it be any table or desk in that room, let it be your airflow laminar hood. Everywhere you have to keep only the products and items and things that are essential and mandatory for your work. Unless and until there is no work for it, that particular item should not be present in that area. So it will cause a lot of clutter. So your clutter means it will get crowded, and then even if you turn on your UV or you try to use your filters for sterile air, or you can spray aerosol disinfectants. When there is more clutter, there is lesser surface that will be covered. So if your entire surface has to be covered during disinfection, and also it will prevent from formation of dust, collection of dust in that area, so use very minimal things which are absolutely mandatory for your work. Next, so correctly label reagents including flasks, media, and ampules with contents and date of preparation. This is also a very important point. So whenever you do something, use something, if you are using it from the media, generally we will not use or pipe it out anything directly from the main bottle. So to avoid or decrease the possibility of contamination, what we do is we will aliquate it. So if you have a main big reagent bottle, if you want to use it, depending on your usage, if you want 100 ml, 200 ml, 50 ml, depending on that, you will collect it in a smaller, sterile, autoclaved tube or container, and then use it from there, and your main bottle will be safe. Even if your aliquot gets contaminated by some accident or some error, your main stock will still be free of contamination. So when you aliquot such things into a fresh tube or a sterile tube, you should always label what it is exactly. If it is a media, RPMI media, label RPMI. If it is a complete media, label it complete. Or if it is incomplete media or serum free media, label it that, and write the date on which you have aliquoted it. So it will be useful for you to troubleshoot any of the contamination, or for the next time of the usage, it is always easier. And ampules are the small tubes, microcentrifuge tubes. And if you are preparing some sort of dilutions or some sort of um, mixtures and some drugs or some reagents of some specific concentration, so it is always important to write the concentration of the reagent or drug or anything that you have prepared, or what are the contents you have used to make the mixture. So all the contents, whichever is required, is to be written on the tubes. Next, only handle one cell line at a time. This common sense point will reduce the possibility of cross contamination by mislabeling, etc. Okay. It will also reduce spread of bacteria and mycoplasma by the generation of aerosols across numerous opened media bottles and flasks in the cabinet. So most of the times we use more than one cell culture lines. So if you are using a single cell line, this will not be a problem, but most of the times we do use more than one different cell. So when you do such thing, it's always better that you maintain a different uh, culture room, or at least a different incubator for another cell line. So if you do this, some cell lines are more susceptible to contamination, some cell lines are not much susceptible, and even if they are like equally susceptible, or you don't know which one will get contaminated. And there could be some cell lines, like we spoke in the last session, that you might have to first proliferate them in one temperature and then differentiate them in other temperature. And then there are cell lines which you directly proliferate and then perform your experiments continuously. So in those cases, what happens, even if your cells get contaminated, the cells which are in continuous proliferation and usage state, you can throw away and then take a fresh vial after decontamination. It might take a week or 10 days. But when you are using cell lines which need more number of days for their growth, and then more number of days for their differentiation, and now this cell line is contaminated because of your some other cell line which can be reproduced easily, but then this cell line needs some 14 days for itself, 20 days for itself to differentiate. So such cell lines, it will be difficult for you to again stop the culture and then bring a fresh one and revive, and again you will lose a lot of time in this. And other than this, first of all, it is very difficult to identify a contamination. So sometimes you might not know that there is contamination, but you might get varied results. You do not know what is happening. Like in case of mycoplasma, unless and until the mycoplasma is spread completely in your plates, it has grown vigorously, you cannot identify a mycoplasma contamination. So for such cases, it is always important for you to maintain different incubators at the least for different cells. So next is clean the work surfaces with suitable disinfectant. Example, 70% ethanol between operations, and allow a minimum of 15 minutes between handling different cell lines. So as I said, when you're handling different cell lines within the same cell culture, you most of the times we cannot afford to have a different cell culture for a different line, so you have different incubators, or we also have segmented incubators wherein there is one hole, like okay, so your basic incubator will be like this. This is your incubator and this is your door. Okay, so here you will have racks, 3 or 4 racks depending on it, and here you'll have a space for your water. So this is your basic structure of your cell culture. So this is your common glass door, this one. So in such kind of case, you cannot separate the segments and you cannot use different cell lines within the same incubator. But whereas in there are other incubators wherein you will have segmentation here. They are called segmented incubators. So this is your one segment, this is your another segment or compartment within your incubator, and this is your another segment, and each segment will have an individual door. So unless and until you open that door, the probability of contamination between this to this segment, this to this segment, this to this segment is very less. So in those cases, you can use a different cell line here, you can use a different cell line here, but at the same time you have to remember that before you start working on a different cell line, that is now you have first seeded, split, or treated cell line A. So now immediately you want to do cell line B, but you can't do it immediately. You have to give a minimum of 15 minutes gap between your cell line A and cell line B, and in between you have to again sterilize, using a disinfectant. You have to turn on your UV in the hood and then start your cell line B. Okay, and you have to use a suitable disinfectant. So there are many disinfectants available. Most standard disinfectant that we use is 70% ethanol. We uh, we dissolve 100% ethanol in water, 70:30 ratio, and make 70% ethanol. Or else you can also use cell biology disinfectants which are available in the markets. So there are products, because ethanol is a skin irritant. So if you try to sterilize your hands, and if you try to sterilize before wearing gloves, if you use ethanol for a couple of days, you might be okay, but then after regular use of it, your skin starts to dry, and you get this dry skin condition, and then it causes irritation and some other skin condition. So nowadays they have started this skin friendly disinfectant which are available. They are also effective, but 70% ethanol is most effective. And you can also use isopropyl alcohol, that is also there. So depending on your requirement and usage, you can choose your disinfectant. Next is, wherever possible, maintain separate bottles of media for each cell line and cultivation. So this is also again going back to maintaining different cell lines completely as a different set. So if you have two cell lines which have a same media, which use same media like DMEM, so you will use one bottle for cell line A and a complete new bottle for cell line B. And you will also write it down on the bottle that this is for HEK, this bottle is for HeLa or HEG, something like that. So as to make sure that by the exchange of media also you don't contaminate your cells. And next is, examine cultures and media daily for evidence of gross bacterial or fungal contamination. This includes medium that has been purchased commercially. So, whenever you work in the cell culture room, whatever you're working on, either your plates, your media, your reagents, anything that you use, you have to always check for contamination, either by some kit before starting a new cell line, contamination kit, or you can even while subculturing your plates, you have to check your plates under the microscope for signs of contamination so that you can maintain healthy cell lines. Next is quality control all media and reagents prior to use. So you have to make sure that all the media that you purchase and all the reagents that you add in the media that you purchase, like FBS, or the enzymes that you use like trypsin for detaching the cells, all these things you have to make sure that the company has quality checked, that there has been a quality assurance certificate for that, and then only you can trust those media and buy them. And when you there is also a way to prepare your own media by using media powder, lyophilized media powder. So that time you have to make sure that you yourself are making this quality check at every step of media preparation. Next is keep cardboard packaging to a minimum in all cell culture areas. So whatever packaging you get, your cell culture dishes, cell culture plates, your pipettes, your serological pipettes, your tip boxes, your tips, everything, it is always better, and it is practiced that you remove the uh, packed items from the cardboard box and you stack them in your cupboards or in your drawers in your cell culture room, which are already sterilized. So because cardboard boxes, you cannot completely sterilize them because they travel outside and come, and the packages inside are completely sealed, so it's always better to take them out and instead of putting your entire cardboard box inside your sterile room. Next is ensure that incubators, cabinet, centrifuges, microscopes are cleaned and serviced at regular intervals. So everything that is present in your cell culture room has to be cleaned regularly with your disinfectant, whatever you use. All the cabinets, all the centrifuges, the refrigerators, if you have it inside the room, the laminar airflow outside, outside of the incubator, everything has to be cleaned at regular intervals. Last is test cells for mycoplasma on a regular basis. So first you test your cells for mycoplasma when you get it from your company or your source, wherever it is, and you see that when you start your cell culture itself, you did not get a contaminated cell line. So you will know the difference between a contaminated and non-contaminated cell line. So the cells which are supplied by companies like ATCC, etc., they also certify, they do the mycoplasma test on the cells, and they give us mycoplasma contamination free certificate for their cells. But if you want to take an extra step, you can also there is a kit available for mycoplasma detection. So you can buy that kit, you can test your cells before seeding and before starting your cell culture, and also like at regular basis, like once in 6 months or once in 1 year, you can do this check test again. Okay, if anyone has any doubts, you can always ask me at the same time. Okay, next, so now we go to don'ts. So what is your first don't? Do not continuously use antibiotics in cell culture medium, as this will inevitably lead to the appearance of antibiotic resistant strains and may render a cell line useless for commercial purposes. So you know the contents of media, so you use antibiotic in media, usually pen strep, that is penicillin and streptomycin. It's a mixture for both, it's a cocktail for both bacteria and mycoplasma or fungal contamination. So most of the times what we do is When you start a cell culture and when you grow it, you use the antibiotic. But when you want to, like use it for your experiments, sometimes the antibiotics may not be or may be an obstacle in your experiment, or you might not want the effect of the antibiotic, which may or may not be there, but you still don't want to take that risk. You do not use your antibiotic in your experimental plate. And also, when you are culturing continuously some cell line, if you like some passages of cell lines which goes for a long time, like some 20-30 passages, so if you use antibiotic throughout this, so it might take some 2-3 months for you to do this 20-30 passages, and all through you have been using antibiotics, and you know that even if you take all the measures and you do all the steps required for sterilization, bacteria and microorganisms are omnipresent. So there will always be some kind of entry of bacteria in your area, and if any of these bacteria are resistant to it, or they have, they will attain the capacity to become resistant to that antibiotic, because you're continuously growing them in that, so they will gain that capacity to survive in that place. So it's better that you go alternating between on and off with your antibiotics. So once you get an antibiotic resistant contamination in your cell lines, then you cannot eliminate it. Whatever you do, your cell lines will be contaminated and it will become useless. Next is don't allow waste to accumulate, particularly within the microbiological safety cabinet or in the incubators. So as I previously said, do not clutter and don't accumulate your discard. Your waste here is your discard, like you uh, remove your media. When you change your media, you remove your spent media, so immediately you have to discard that after your work is done over there. Don't leave the discard inside your laminar hood or in the room or anywhere. So it's better to, there is a dispenser for discard which most labs, sophisticated labs use. So the discard will always be outside your incubator. It will have a fine tip, and then the tip, like your normal tip with which you pipe it, and the tip will be connected to a tube. The tube will go outside your hood and it will be collected in your waste bin. So even that you have to discard immediately. Do not leave it. So what you do sometimes is, if you're not having the dispenser, you pipe it out into your discard beaker or bottle, and then you add hypochlorite in it, which will immediately dissolve all your contaminants. And then once you're done with your work inside, immediately go and add more hypo, make sure there is nothing left in that, and then discard it. Next, uh, don't handle cells from unauthenticated sources in the main cell culture suite. They should be handled in quarantine until quality control checks are complete. So this is when you get your fresh set of cells, from your main source, either that has been gifted from some other lab or you have got it from some commercial vendor. Always handle them in a controlled environment. Do not handle them openly where you are already having some cell lines in running, and don't mix them with them, because you don't know you're getting this for the first time. It may have already been contaminated, or it may have contaminated during the transport, during spillage, leakage. It could be anything. So first, when you start your new cells from a source, always do it in a controlled environment, in a quarantined environment. And unauthenticated sources is like, sometimes you might borrow cells from someone just to see something, just to check something, without official transaction. When you do that, you have to always be handling it in a separate area. Next is avoid keeping cell lines continually in culture without returning to frozen stock. So this is what we were talking about, the continuous subculturing, which is like the range of passaging. So when you culture, you go to particular passage number and then you freeze them and then start a new vial. So this cycle has to be running. And then when you start a new vial also, you will start with the oldest vial. It's not like you froze a plate in a cryo vial 2 days ago and then after 2 days you want to again revive a new vial. It doesn't make sense for you to again revive the same vial which you froze 2 days ago. So you go to an older tube which you froze few days ago or few months ago and then revive that. So in that way your cells will not get your stocks will also not get old in your freezer. So you will always have this cycle of good and healthy cells running if you do this. Next is avoid, and your frozen stock you have to also sometimes again start with your main frozen stock. Like you got your fresh source of cells and then you so immediately what you do, you don't just keep subculturing them. You subculture it once and then you freeze some number of vials in your first set of your source, so that always you have a chance to go to the original source and revive the cells. Next is avoid cell culture becoming fully confluent. Always subculture at 70 to 80% confluency or as advised on the ECACC cell culture datasheet. Whatever the company's datasheet is given, they will always say which is the optimum or the maximal confluency this type of cell has to reach. So any type of cell, you should not let them grow or proliferate more than 80% confluency. We spoke about the confluency concept in the previous session. So if anybody uh, does not know this, you can ask me, I will tell you again. So when your cells are 80% confluence, you have to either treat them, that is use them for your experimentation, or you have to split them and subculture them again immediately. So you do not let them go beyond 80% confluency. That is always help for maintenance of healthy cells. Next, do not allow media to go out of date. Shelf life of shelf life is only 6 weeks at 4 degrees once glutamine and serum is added. So yeah, so most of the times we do not get the media bottles as soon as they have been produced, because they have to be imported and then they have to be transported. So few months will be lost in that. So always see that you have a minimum of uh, 6 months expiry left in your product. Like for example, for medias, usually the expiry will be somewhere around 1 year or something. So sometimes you might get a product which has an expiry date of next month or in 2 months. You might not use, because one set will have some 6 bottles minimum, so you will not be able to use 6 bottles within 1 or 2 months, unless like there are many people doing it. So you will not use those many. So see that you ask your vendor to supply you a newer product which has at least a minimum of 6 months expiry date on it, so that you will not use expired products. Because mainly amino acids, serum components, once they are in the media, they will start their process of degradation, and after their expiry date you might not even know whether these things are still present in your media or not. Serum, however, we add it, so you know the date you add the serum into the media. But glutamine, L-glutamine and other amino acids, vitamins, these are all pre-mixed in the media, so it's always better to check your expiry date. Avoid water baths from becoming dirty by using, okay, this is a product, this I have taken from Sigma, so this is for promotion of their product, I'm not promoting it. But the point is, avoid water baths becoming dirty. So when you use water baths, you have to always replenish the water, discard the old water, clean it again, wipe it with disinfectant, alcohol, and keep this under regular check, because this is where there is maximum possibility to get contamination from. So because of this risk of contamination in water baths, many people keep the water bath out of the cell culture room, or many people do not use water bath. There are newer heating baths which have metal balls in them, so the heat is dissipated via the metal balls and there is no use of water. So those are like absolutely free of contamination. But most of us still use water bath, so always see that your water bath is clean, not contaminated. Once you after use, see that it is properly closed and maintained in a proper space, sterile space. Then don't allow essential equipment to become out of calibration. So ensure microbiology safety cabinets are tested regularly. So when you have some equipments that you have been using, uh, like your centrifuge or some other equipments, like I told you that sometimes we prepare our own media. So you have to see that whatever equipment you're using uh, in the cell culture should be calibrated, cleaned, maintained in a very proper condition all the time. So these are the main things that we have to follow. So these are the do's and these are the don'ts. So if anybody has any doubts in these two slides, please let me know. Anyone? Okay. Uh, okay, if there are no doubts, I will continue. I hope the slideshow is still visible and I'm audible. So next, so this is your layout of your cell culture room. So as we saw in your weekly syllabus, the design layout was there. I hope it was all, you have watched all the videos for it. This is just a basic outline of how it should be. So I will tell you what is BSL 1, 2, 3 also shortly. So first, you in a well maintained BSL facility, you will have your this first area where you have your shoes, your lab coat, your at your entrance itself, and then there will be a blower. That area will be closed from all the other areas. And after that, you will have an area where you need all these extra things which are not directly connected to your culturing techniques, like your water bath, your chemical. Like I said, water bath will be usually kept in a different area other where your laminar or your incubator is not there. So your water bath, your discard sink, this is the sink where you only discard the things from your cell culture room, nothing else, nothing else. And then your fridge you need for storage of all your reagents, media, your liquid nitrogen tank, and then the area where you sterilize some things, you add your hypo and all these, and then your chemicals, like your cabinets basically, where you store some things that are useful. And then you have your main area where you actually perform your cell culture experiments or techniques. So here what is absolutely necessary is first your CO2 incubator, and then which is connected to a CO2 tank or a cylinder, and then a laminar flow hood, laminar air flow hood. And then you need a centrifuge because you have to pellet down your cells, you have to resuspend them, so you need a centrifuge. So you cannot always like go out of your room to find your lab centrifuge, common centrifuge, and it might be contaminated, it might not be sterile. So a centrifuge is always in your sterile space. And then you have your microscope for your observation of your cells, your regular check for your contamination of microbes, etc. So usually these are the also the layouts, the distance that is given for a general lab, that this should be the distance for this should be the distance. Next, we will talk about biosafety levels. So in biosafety levels, which is nothing but BSL, you have 4 BSL levels. So first is 1, 2, 3, and 4 respectively. So biosafety level 1 means it's a risk group 1. Like this is basic research labs. Most of the basic research labs are biosafety level 1. One second, excuse me. Excuse me for a minute, I'll be back. Okay, so where were we? We were at biosafety levels. So in your biosafety levels, the first level is your basic laboratory research lab safety level, where you use agents like E. coli, you do transfection, you do basic cell culturing, and you have this, like even at your master's level you might have done internship. So those labs are all this biosafety level 1, where your basic teaching happens or basic lab research happens. And then what is the laboratory practice for this level is GMT. Does anybody know the full form of GMT? Am I audible?
Good manufacturing practices.
Close. So it is good microbiological techniques, or basically good lab practice. Okay, so the do's and don'ts that we had, or in the first session when we talked about all the terminologies to know what is what. So all this comes under good lab practice or good microbiological techniques, where you do everything under controlled, regulated, sterile conditions. So that is enough for this. And then what safety equipment you need for this is, because you're not dealing with anything that is dangerous or infectious, at your immediate effect. You don't really need much of the safety equipments. Only your, even in your open bench you can work, like in your MSc labs or in your bachelor's labs. You can do your basic, with a burner, with a Bunsen burner. You can inoculate things, or you can uh, pour your plates, heat your agar, that sort of things. But even that area should be disinfected. That is your basic lab practice, your proper disinfection of areas. Not talking while inoculating or making media, not breathing, not singing, humming things. Okay, so that is your good lab practice or good microbiological techniques. So that comes under your group 1. Now, what is group 2? That is your BSL 2, is which can come under your primary health services, diagnostic services research. You might have gone for your diagnostics for a blood test or something. Sometimes they collect your blood tests in the lab or near the lab. You might see that the lab is within the center. So those all comes under 2. And at the same times, your research labs, where we, have these cells from patients or specimens, or even animal specimens, and some not so dangerous viruses, because all viruses are dangerous, but these might not be this high level dangerous virus. So like herpes virus, which do not spread other than their original route or something like that. So when you are dealing with these kind of things, you have your BSL 2 facility. So here what you have to do in your lab practices is your GMT. Your GMT will be there everywhere. That is your basic. Without GMT, you cannot start cell culture techniques, anything, even bacterial, anything you cannot do. So along with your GMT, that is your good microbiological techniques, you have to have protective clothing and biohazard signs, because you're using your human samples, uh, virus, etc. So along with your this safety equipment and your protective gloves and your clothing and your eyewear, you need biohazard signs. So you need to discard. When you discard something, you should not dump everything in the same bin, or you should not dump everything together and then just send it for discard or autoclave. So what you have to do is you have to segregate your discard also based on its level of hazard to the environment, to the society, and outside. So, your animal waste, your human sample waste will go in one hazard, your bacterial waste will go in one hazard, your agar will go in one hazard. So, like that, you have different hazard discards, and for each thing, you have to label it properly, because when it further goes to your, emptying it from your lab, everything will be pooled, and it should not be again, uh, put in a place where it does not belong. So your safety equipment that you need for a BSL-2 is you need to have that bench, and along with this bench you need to have your BSC for aerosols. Does anyone know what is the full form of BSC? Like this BSC and GMT go hand in hand. Anyone? Okay. So BSC is nothing but biological safety cabinet. So when you are working with human derived cells, patient derived cells, virus, you cannot do it in open, right? You have to do it in a safety cabinet. So your laminar airflow also comes under your biological safety cabinet. So this will contain whatever you're doing within that cabinet, and it does not spread it. And your aerosols, your disinfectants, your UV, the cell culture room might also have a UV lamp. Before entering it, you might turn it on, or after your work is done, you might turn it on, so that it sterilizes the room. Next is your group 3, that is biosafety level 3, where you use these infectious pathological agents which can be spread easily. So it's like when you're dealing with HIV, many people work on HIV and their research labs, and then there is M. tuberculosis or any other, infectious bacteria or fungus or virus which can be spread via the air, that comes under biosafety level 3. So this is special diagnostic centers where they work on this, and special labs which uh, have the license to work on this. So when you have to work on such high risk virus or pathogen, so you need to get a special license for your lab. Unless and until you have a biosafety level 3 cell culture, you cannot get this license. So it's like that. So then, what are the lab practices here? This comes under level 2 lab practices. So special clothing, definitely you need to wear your masks, not only your eyewear, headwear, gloves. You need to also wear masks, because some of these pathogens, if you inhale, you might get infected. So you might wear your PPE kits also. So you need to have access control here. So this is very important, because maybe in 1 and 2 this might not be a bigger issue, but many labs do have access control in 1 and 2 as well. Maybe not in 1, but 2 they might have. But maybe that might be to protect their IP, like their intellectual property. But here it comes as a responsibility, because you're dealing with highly infectious pathogens. You should not let anybody enter that room unless they have access to it. If you are a trusted known person who is known to be working in that, then within the lab also, you should get the access to enter it. Only then you should be allowed. Not all lab members might be allowed. Maybe who is not working in that area is not allowed. And definitely anybody outside your lab should not have access into your BSL 3 facility. And there should be a directed airflow, like your cell culture room will be ventilated in such a way that the airflow from your lab will go into your filters, and then it will be, the air also will be treated. Because if by mistake some breakage, some spillage has happened and it has spread within the room, it has to be either contained within the room and be, treated on, or it should, the air that goes outside your lab facility should go in such a path that it does not contaminate the entire atmosphere. And your safety equipment is your BSC, that is your biosafety cabinet, very important first, and then your primary devices for all your activities. That's what the airflow, your access control, your even your laminar airflow will be different for it. It should not come directly towards you. The airflow will be directed within the laminar hood towards outside your outlet. Next, and the last one is your biosafety level 4. So these are like very highly sophisticated uh, culture rooms, culture laboratories, where you work on this highly infectious and highly dangerous and, life uh, life taking pathogens, like your Ebola, your Marburg, your COVID, your Lassa, your MERS virus, and this recently the Mpox virus. So these kind of uh, pathogens which are high risk to your life, and maybe there is no treatment for it yet, or you are creating a vaccine for it. So all these comes under your BSL 4. So these are dangerous, like extremely dangerous pathological units. And here you have your level 3 plus airlock entry. Even the air that comes inside, the air that goes inside has an entry box, has a treatment that will happen to it. And then you will have a shower exit, you will have a blower, you will have a shower exit. After working in that lab, you have to, get yourself cleaned again before coming out. And then there is a special waste disposal. You have to incinerate all the things that you use. So high level of maintenance has to be done for this. So your class 3 BSc, your positive pressure suits, your class 2 BSc is double-ended autoclave. So everything that goes inside, comes outside will be sterilized, autoclaved. Unless it's a reusable material, you will incinerate it. So this is your BSL 4. So now we saw what is BSL 1, 2, 3, 4. You can also go and check which organism or which pathogen comes under which BSL. You can check for labs which have these facilities. Okay, so now, yeah, like I told, GMT means good microbiological technique and BSc means biological safety cabinet. Now after this we will be going for the quiz or the questions. So till now, if anybody has any doubts, you can go ahead and ask me. Anyone? Hope you all are listening. Anyone? Any doubt in all this introduction, your biosafety levels, your layout of your cell culture room, what all is required in your room? Anything in this? Or if anybody didn't know some terms that I might have used, if you want to know what that is exactly, like your confluency or your anything else. Okay, so if there are no doubts, we will continue. Okay, hope the slides are visible. Okay, so this time, last time I quizzed you with the same questions of the assignment, but this time I wanted to not give the same questions because it's not like it should not be a spoon feeding. So this time I have prepared some questions which are similar to those. So if you do this, you hopefully might be able to do your assignment on your own. So let's start. So now you have to answer the questions, okay? So you can't be quiet. You have to tell me some answer, whether you know or you don't know, or give an answer to the question. It should be interactive, okay? So the first question is, in culture media, dash is used as a buffering system. Your first option is sodium carbonate, second and CO2, and your second option is phenol red, third is HEPIS, and fourth is both A and C or A and C. What is your answer? Anyone?
Option D.
Option D, very good. So both your sodium carbonate, carbon dioxide, and your HEPIS, both are buffering systems. So your answer is option D. Very good. Clap for yourself. So here you can see, like, I have put some basic components of your media and what is their function and some examples for it. So here you can see in your buffering system, the role of your buffering system is regulation of pH. So here you have some natural buffering system like the CO2 gas, which balances the buffer along with your bicarbonate. And then HEPIS, which we spoke here, is a buffer. It's a chemical buffering system, and it is more superior to your natural buffering system, and it does not require an external gas from the environment. So your basic pH which it has to maintain is your range between 7.2 to 7.4. So 7.4 is your basic buffer pH. So this will have the capacity to maintain that. And other than this, you can see next is your pH indicator, which is the function of it is to detect the variation in pH, like the change in pH. It will let you know. Example is your phenol red. Then inorganic salts. So salts, inorganic salts like sodium, potassium, calcium ions, these are also present in your media. So these help in maintaining your osmotic balance and also your membrane potential, either if it is your cells which require to maintain your membrane potential, or your uh, cellular organelles like your mitochondria, etc. So it will help in maintaining your membrane potential. Then amino acids. Amino acids like glutamine, I think we just spoke about glutamine. So the L-glutamine is definitely always there in your media. This is important for your synthesis of proteins in your new cells. Now, when you give the media, the cells will proliferate, or it will grow in size, or it will differentiate. So, for this, it needs new proteins. It will synthesize new proteins after activation of genes. So, that time it needs what is your raw material for protein synthesis? Your amino acids. And it also provides nitrogen for NAD, NADPH, and nucleotides, and serves as a secondary energy source for metabolism. So you also know that other than sugars, that is, other than carbohydrates, proteins, amino acids are also a part of the metabolism. So you also get energy from your proteins. So it's in that way, amino acids are very important. Next is your carbohydrates. These are the main energy source, that is your glucose and galactose, and some might contain maltose, fructose. So depending on the media and depending on the cell type, requirement of the cell type, your experimental design, your treatment, drug experimentation, your carbon energy source might differ. So glucose is the most standard energy source for as a carbohydrate. Other than that, galactose will be there, and some might contain others. And then you also need fatty acids and lipids, because your cell cannot survive only with carbs and proteins like your body. So cell is nothing but an point-sized version of your body, right? So your body also requires fatty acids and lipids for its normal functioning. So they're very important in serum-free media, as they are generally present in serum. So generally we add this, like I told you about this complete and incomplete media, right? So in your complete, your usually whatever media you purchase from the companies, it will be a incomplete media. That is, it will not have serum. You have to add serum after you get the media, and it will have a percentage. Usually we add 10% serum for proliferating cells. So when you use incomplete media, And this serum is a rich source for fatty acids and lipids. These are very important for proliferating cells. And when you use incomplete media, if serum is not required either to stop the proliferation or to when you are using some reagent or you want to check activity of something on the cell, and your serum components might uh, interact with that reagent or that drug or that enzyme or whatever that you're checking the activity of. So that time you use incomplete media to give this treatment in. So that time also the cell has to be healthy. So the media itself will also contain a trace amount of fatty acids and lipids. And other than this, vitamins essential for growth and proliferation. It's very important. And then trace elements like which are needed in like very tiny amounts, maybe in like nanogram or microgram scales. So these are micronutrients which maintain the activity or function of an enzyme, like copper. These are coenzymes, cofactors. All these are, they act as, so like copper, zinc, selenium, tricarboxylic acids, etc. So these are your trace elements. So your answer was D, both natural and chemical buffer system. Next question is, which of these is a sterile place to seed your cells? Lab bench, open room, laminar flow hood, incubator. Where will you seed cells or inoculate your cells?
Ma'am, laminar flow hood.
The laminar air.
Yes, very good. So your answer is C, laminar airflow. So you should also know that lab bench also you can do it like your BSL-1 facility, but it's not completely sterile. You still have the risk of contamination. So your most sterile place to seed your cells or inoculate or subculture is your laminar airflow hood. So your laminar airflow hood comes in two types, like I said before. One is your vertical laminar airflow hood and your horizontal laminar airflow hood. So this vertical and horizontal refers to the direction in which the air flows inside your hood. So here you see a vertical hood. This is your workstation, your workbench. This is your glass door, and you will be here outside, sitting here, and you will be facing towards this glass door, and you will be working here. Your hands will be here. So your airflow is coming from top. So there will be a suction, there will be a fan which will pull the air from top and then send the air inside the hood. And from inside, the outlet is this direction towards you where you're sitting. So the vertical laminar flow will go towards you outside. Okay. And then you see like a rotator, like a fan, etc. here. And then there is horizontal airflow. So in horizontal airflow, what happens? The air flows in this horizontal direction. I hope you guys know what is vertical and horizontal. So here there is one more thing. So here in this particular picture, it's showing as if it's coming out directly. But sometimes, most of the times, what vertical has is it goes like this. Here there will be a separate system where they will have more filter pads and the air will flow out through that. But in the horizontal, what happens? The air comes from this back side. So you are sitting here, you're doing here. So air is directly flowing at your face. You get it right, the direction. So this comes down and then this goes out, and this comes directly from back to front, kind of left to right sort of thing, from here to here. So this is your horizontal. Next question is, which component of the laminar airflow ensures sterile environment? A. HEPA filter. B. Fan. C. Blowers. D. Stainless steel body. What is the answer?
Ma'am, HEPA filters.
Yes. So it is your HEPA filter.
HEPA filter.
So what does your HEPA filter do? So your HEPA filter will filter the minute particles in your uh, airflow which comes, like you saw in the previous, your air is flowing. So your HEPA filter is here in this one, and here your HEPA filter is over here. So when the air is coming inside the hood, it will be filtered by the HEPA filters. You can see it, this is a laminar airflow hood. So here you can see many components of this. So this is your UV lamp. Many of the hoods come along with the UV lamp for extra sterilization. This is your LCD control. It will tell you what is the air pressure, what is the number of hours UV has been on, etc. And then you will have your HEPA filter. So is this your horizontal or your vertical? Anybody?
Ma'am, it's vertical.
Okay. Why? Why is it vertical? How are you saying it is vertical?
No, ma'am. Sorry, ma'am. It's horizontal because the HEPA sheet is parallel to our face.
Yeah, so your HEPA is over here. So this is your horizontal. So your HEPA might also be put here on the top instead of this. So this is both. You can use it either way. So here on the top also you can have your HEPA filter and your air will flow like this, and then one outlet can come like this. Okay, they also have this primary efficient filter. So if your air goes like this and comes out like this from your filter, then it is your vertical. But if they are saying that the HEPA filter is here at the back, then it will come directly outside, then it becomes a horizontal. So what I'm trying to say is the build will be similar to everything. How you should see whether it is a horizontal or a vertical is where the filter is placed. So wherever the inlet is coming, there that is the direction of the airflow. And then you have your stainless steel working body, and then you have your caster, you have, that is your wheels, if you want to transport, move it to a different lab, or if you're shifting, it's convenient. And then this is your safety glass, and then that's it. These are the main things. uh, why do we use stainless steel in the working area? Any idea? All the laminar airflow will have stainless steel only. Why can't we use something else? Anybody? You can guess. Why do we generally use stainless steel?
Ma'am, might be it is because it is junk proof.
Junk proof. Yeah, like what do you mean is like rusting, to prevent rusting,
Y
right?
yes, ma'am.
Yeah, so usually whatever chemicals or liquids, reagents we use might cause erosion, or if your moisture or something has fallen and all, it might cause rusting. So stainless steel will prevent your rusting and dust formation inside your hood. So HEPA filter, the high efficiency particulate air filter, is present within the cabinet that makes the environment more sterile for the operation. The pre-filtered air passes through the filter, which traps fungi, bacteria, and other dust particles. The filter ensures a sterile condition inside the cabinet, thus reducing the chances of contamination. Okay, this is your use of the HEPA filter. We'll go to the next. Next one is liquid nitrogen is essential for which step? Thawing, seeding, subculturing, freezing. Tell me.
Or freezing.
Yes, so you need liquid nitrogen for freezing cells. And how do you do this? You take your cells, you pellet them down, and then you suspend them in freezing medium, and then you aliquate them into cryogenic vials. So you cannot use any regular vial also, or any regular centrifuge tube or microtube or ampules to freeze it, because you're putting them in liquid nitrogen. The material that the vial is made of should be uh, adapt or appropriate for transferring it into the liquid nitrogen, and it should not get damaged. If it gets damaged, leakage and then contamination. So you should use cryogenic vials which can tolerate that extreme temperature of -130. Then you place the vials in a controlled rate freezing container. So this is also one very important thing. So when you put it in the liquid nitrogen, before putting that, what generally we do is we freeze it to -80. We do not like flash freeze it in the liquid nitrogen. We first place it in a container like this, which will have isopropyl alcohol, or there are also some newer versions of this container coming which does not even require any alcohol. The main function of this container is to decrease the temperature in a controlled rate. That is, it will decrease -1 to -3 degrees, like 1 to 3 degrees per minute, so that this formation of liquid crystals or solvent damage, solute damage to the cells does not occur, and your cells also does not get any shock. And then you put this in the -80. So in the -80 also, if you put, it will freeze quickly because it's obviously -80 degrees inside. So you put it in this container first, and then you put the container in this -80 fridge or freezer. So per minute it will go down by -1, -1, -1 degrees. After that, maybe after a day or two, you can shift this tubes from this cryo uh, container into the liquid nitrogen tank. So this is how the tank looks. You will have liquid nitrogen in it, and these are the racks which you will put inside this tank. So you see they have this long handle. So this will go till here, and this handle will be here. You will pull the handle to get all the racks out. And then you see these green, bluish green kind of things. Those are your 96 well boxes where you can put your tubes. You have this box which has this 96 compartments where you can put your this cryo vials. So you put your vial in one of the tubes, you put the tube in the rack, and you should remember, when you're using it commonly in the lab, you should remember which rack is yours. You have to label it here, and then in which rack, in which shelf number you have put your box. Usually one box will be for one person, so you should remember, in like, if this is the third rack, in the third rack I have put my box in the second shelf. So you should note it down somewhere or remember it. So next time you go, you can't open it for a long time and search it everywhere. So you should know exactly where your tube is when you want to take it out. So you should remember and note this place and then put it inside. Okay. So when you're taking all this out, you should remember what all you have to wear. You have to wear your protective gloves. You have to wear your goggles, safety goggles. You have to wear your mask if you have breathing trouble. All these things, and splash proof apron because it's liquid nitrogen. So storage, why do you store it in liquid nitrogen? Liquid nitrogen gas allows the lowest possible temperature to be maintained with absolute consistency. So it will maintain this temperature of -130 at a consistent level. So you may not worry about if power is lost, my samples might melt and thaw, or if any other problem is there. Unless and until your tank has liquid nitrogen, it will maintain at -130. So your cells will be safe and it will not get affected by change in temperature. Then mammalian cells are cryopreserved to avoid loss by contamination, to minimize genetic change in continuous cell lines, and to avoid aging and transformation of finite cell lines. So if there is temperature change in your stored cells, if they've thawed, they're frozen, or some kind of disturbance has occurred, so all these kind of effects you can expect. So it might have undergone any genetic change, or it might have few of the cells might have died, few of the cells might have transformed to something else, some weird kind of morphology, or few of the cells might have aged. So to avoid all this, you have this consistent temperature maintaining liquid nitrogen. Okay, so next question is, alcohol is used for what in cell culture? Storage, disinfection, centrifugation, resuspension.
Ma'am, it's disinfection.
Yeah, so you use alcohol for disinfection. So this alcohol also, you get alcohol from your vendor, and you have to use it very rationally and very use it only when it is necessary. You should not, spend it overtly, saying that if I wipe it 10 times with alcohol, it will get 10 times more cleaner. It's not like that. If you have made a proper proportion of your alcohol, proper 70%, or if you get directly 70% bought, if you have spread it across your bottle or your surface evenly everywhere, and then you wipe it out after the appropriate amount of time, once is more than enough. You don't have to do it like 100 times to have this kind of surety. So if you do it once itself, do it perfectly so that you don't waste it, because this they won't supply you a lot. Usually what happens, your institute and your universities will get this contract for alcohol supply, and it will all be licensed, and because it's alcohol, right? You cannot just buy it randomly. So your institute will supply it to each lab. It will give only like 5 liters to one lab for one semester. So you should not waste it, because nobody else will supply it to you if you have finished all of it. So use it very rationally, very, consciously. Mode of action: how does alcohol disinfect? Yeah. So do you know how alcohol disinfects? Anybody? Do you want to tell me the answer? It's given here. You can also read it. Okay. So when you spray alcohol, it's not like you spray it. It's not like your regular Colin or some, Lysol or something. You just spray it and you wipe it off. You have to spray it and wait for some time, because how your alcohol works is it dehydrates the things. It evaporates, right? It's volatile. So it dehydrates your cell. So when your cell gets dehydrated, only then it will die. If you just spray it and you wipe it, it might not be that everything has been disinfected. There might be some area where your alcohol did not fall, and then you wiped it, that area is still dry. So don't do like that. Spray it properly, wait for some 30 seconds for it to dry a little bit, and then whatever remaining is there, you can wipe it out. So it dehydrates the cell, disrupts the membrane, causes coagulation of protein. So this is how your cells or your bacteria, your fungus or anything will die because of alcohol. So your examples for alcohol is ethyl alcohol, that is nothing but ethanol, isopropyl alcohol or isopropanol, and your methyl alcohol. So different things are used depending on where you use it, or your lab practice. So 70% ethanol is used as an antiseptic on skin also. So you can sterilize your hands, but regular use will cause skin irritation. Isopropyl alcohol is used for sterilizing the L-shaped rods when you plate your bacteria, or when you do pour plate technique. So it's like that. And fungus and all these things, you use methyl alcohol, which are more dangerous. So next is, how do cells get oxygen in incubator? Do they get it from cylinders, that is oxygen cylinders, or from cells within themselves, or are they added externally, or does it get dissolved in the media from air? What is the answer? What could be the answer? Anybody? You can guess. Even if you don't know, you can guess. Anyone? Are you there? I want you all to guess all the questions, even if you don't know. Did I give away the answer? Alright. Where do they get the oxygen from? Anybody? I see 25 people, except me, like 24. Anyone? You can guess it right, like anything. You can tell anything you want.
From option D.
B. B for ball. Is it this?
I said B, dissolved in media form.
Dissolved in media. Okay, yeah, correct. So, did you see the answer?
No, ma'am, I just guessed it.
Okay, okay, good. So, your answer is it is dissolved in the media from air. Like for CO2, because you have to have only 5% CO2, we have the cylinders which will have the pressure gauges. It will set the percentage, and you have the pressure valves, and you will have only 5% inside, which will be detected by your detector. But uh, oxygen is directly dissolved from your atmospheric air. How we respire, in the same way they get the oxygen, but the oxygen will be dissolved in the media. They cannot, directly breathe it. So it will be dissolved in the media. There is one more thing I would like to tell you. So usually the oxygen which we get in our body is somewhere around 2% or 2 to 3%. But the atmospheric oxygen level which these cells get when it's dissolved in the media is approximately 18%. So basically, we do not have control for the level of oxygen, like I said. So sometimes what happens, the high oxygen amount can cause this hyperoxia. There are two things. One is hypoxia and hyperoxia. So hypoxia is nothing but low levels of oxygen. Hyperoxia is excess oxygen. So your excess oxygen can also, have some effects on your cell, because oxygen affects your gene expression, it affects your energy metabolism, it affects your mitochondrial morphology, because oxygen is important for your electron transport chain. So your healthy mitochondrial dynamics depends on oxygen levels, your antioxidant levels, your ROS levels. ROS is your reactive oxygen species levels, that is your free radicals. All these depends on your oxygen levels. So then indirectly they also dictate proliferation, differentiation, property of the cell. And sometimes few drugs might be effective only at optimal oxygen levels. So all this comes into play. So how will it cause these changes? So like I said, your low oxygen level is called your hypoxia. Okay. So your low oxygen level is your hypoxia. You might have heard this term. It's a very important hallmark of cancer. So your low oxygen is hypoxia. Your high oxygen is hyperoxia. The pen to write. Sorry. So your high oxygen is hyperoxia. So what happens in your low oxygen? This is just extra information I want to give you. So here in your low oxygen level, what happens is your HIF1 enzyme, HIF1 protein will get accumulated, because there is a phosphorylation which is VHL site. So you have an enzyme which is called prolyl hydroxylase. So what it does is it hydroxylizes HIF1, that is OH. So when there is low oxygen, this will not occur, and your HIF1 will not be degraded. In normal oxygen levels, your HIF1 will interact with your PVHL because of its OH groups, and then this will be subjected to proteasomal degradation, because HIF itself means hypoxia inducible factor. So only if hypoxia is present, this factor has a role to play, because in normal conditions we don't need it. It will be degraded continuously. But if there is hypoxic condition, there is low oxygen levels, then this will not happen. The enzyme will not be functional, and your HIF will start accumulating in your cell. So once the HIF starts accumulating, it has two types. One is your alpha, one is your beta. So both your alpha and beta will now enter your nucleus, and these are transcription factor proteins. So what they'll do, they'll form the transcription factor complex. They'll attach to the area of target genes, and it will start regulating them in a different way. So your glycolysis will change, your proliferation will change, your survival technique will change. So like this, they can change your genetic expression. Same way, in your hyperoxia condition, what happens, there is a increased ROS. First of all, if there is more oxygen, there it will be increase in ROS. So when there is increase in ROS, there are these keep1 proteins. So this keep1 proteins will release NRF2. So this NRF2 also is a transcription factor. So this will go inside here, and it will transcribe a different set of genes. Like here it's a different set of genes, here it's a different set of genes. Here it will start, because there is increase in ROS, it's accumulating, you have to clear it. So it will start producing genes which regulate ROS production, and then some detoxification, those sorts of things. So basically what I want to say is, both less oxygen levels and high oxygen levels are toxic to the cells. So they start changing their genetic expression. Okay. Next is, which of the laminar hoods is better, vertical or horizontal? Horizontal, vertical, depends on your needs, none of the above. What could be the answer?
And horizontal.
Okay. Everybody says horizontal.
Depend on needs.
Depend on needs. Okay. Any other person who thinks there could be some other answer to this? Okay. Someone said horizontal. I want to know, why do you think horizontal is better?
Ma'am, in horizontal. Sorry, ma'am.
It's okay. It's okay. Whatever
Oh,
you thought that could be the
like
answer, you tell me.
I thought horizontal and laminar airflow have HEPA filters, ma'am. But both vertical and laminar air, like horizontal, have HEPA filters, no, ma'am.
Yeah, both of them have HEPA filters.
HEPA filters.
Okay.
That way I thought.
Okay. So someone said it depends on your needs. So that is correct. So both of them are more or less similar. It's only the direction of the airflow that has changed. So depending on, so if you want to know what you want to buy, you should know first how it works. Both of them have everything that is same: HEPA filters, UV. If it is there, it is available in both steel body, everything. Only thing is direction of your airflow. So why you have, and also you need to know why, what type of work you're doing to buy this. But it is also important to choose between horizontal and vertical. It's not like you can buy either of them. You should know what they are and why you should make that choice. So, when choosing a horizontal and vertical laminar flow hood, you must consider the nature of your work and specifications required for your experiment. The horizontal configuration is ideal when you need minimal turbulence in your work surface if you're working with small samples. They are most useful limiting the contamination, if limiting the contamination is your main priority. Like if you're using very small samples, you know what is horizontal, right? This is horizontal. So if you're using very small samples which might get affected by the airflow, air pressure, if the air directly, because you'll be using, if this is your laminar airflow, if your air is coming like this is your door, and your sample is here, okay, and the air is falling directly in your sample, if that will affect your sample, if that will affect your reagent or your chemical, anything that you're using, if it's like, if you're using very minute amounts like micrograms or nanograms even sometimes, so that times this air might damage this, or it might just spread it around in the hood. So that time what happens, you should not choose a vertical one. This is vertical, so that time you should choose a horizontal. So when you choose horizontal, this air will not flow directly and will not give direct pressure on this inside, whatever is inside the tube. So that time you should go for horizontal. And in horizontal also, most of your areas, your the contamination is very less, because from top to bottom equally it will send the air everywhere this way. Okay, and on the other hand, the vertical hood is preferable if there is any concern about the safety of the user being exposed to the samples. If your work involves powders or produces soldering fumes, this could be an important consideration. And if you want to use equipments like spin coater, ozone cleaner in the hood, then vertical airflow will maintain the laminarity around the equipment. So if you are doing some kind of engineering job like this nanotechnology, if you're using making some very minute like chips or circuits, even those you have to make in sterile environment, right? So that time what you can do, you can go for a vertical thing, because your small things, if anything, if you're blowing it this way, it might affect whatever you're doing with those tiny powders or tiny metals or anything. It might just push it out like this outside the hood if you're using horizontal. And also if you're using some infectious samples which can, you know, cause harm to the user who is sitting here. If you're sitting here, if you're using some, you know, virus or some bacteria which is disease causing, and the air is coming from here, so it will directly hit the sample and it will hit you, and then you will get contaminated. So in those kind of cases, you should go for vertical, because vertical, as I said, it will not hit the user, it will go down, it will have another outlet here, it will come out like this. Okay, so you should know what you're doing, you should know what your experiment is, what kind of work you're doing. And then there is if you want to keep any kind of instrument inside in the sterile environment also, it is better if you use a vertical, because that time what happens, if you have an instrument here like this, okay, and the air is flowing from here, so it will spread across this. But you have it horizontal like this instrument will be a obstacle for your airflow, so not all areas will have your sterile air. Okay, that is what they are saying here. If you want more information, I have it in the presentation, you can download it later and have a look. So these are the advantages of your horizontal uh, laminar flow hood, and these are the disadvantages. And then this is for your vertical. HEPA will be in both. Okay, also if you want like to replace your HEPA, you have to replace your filter also, you can't use the same filter for lifelong. So that time what happens in your vertical, it is a little easier to, you know, remove it and fix it, because it's over here, right? It's on the top, you can easily climb and you can replace it. But this, if you want to replace your entire hood, has to be like disassembled from the back side. So even for maintenance purposes, if you know, if you, all the other things which I said in your, in the previous slide, the safety, the infection pathogen, the type of experiment, type of sample, if those are not of much significance, then you can go and think about, you know, maintenance of it, easy fitting of it, that kind of thing. But both have their own set of advantages and disadvantages, so you can read this before you choose to buy which type you want. Okay, so now we'll go to the next question. Storage temperature of culture media, that is your media which you grow your cells in, where will you store that media bottle? Room temperature 4 degrees, minus 20 degrees, minus 80 degrees. Anybody?
4 degrees Celsius.
Yes, so you will store your culture media in 4 degrees temperature. So this is standard, okay? But sometimes it is always important for you to look at the manufacturer's instructions, where they tell you where to store their things, because they know where it will be, you know, uh, where which storage condition will prevent it from damage or degradation. So here you can see two types of media. So this is your RPMI media, mostly used for suspension cells. You can see all the components also written on it: L-glutamine, sodium bicarbonate, and then it is sterile filtered. You preferably do not autoclave your uh, pre-made media. If you're making it fresh, then you autoclave your water, but after you have dissolved all your contents, it's not preferred to autoclave, because autoclave will damage your amino acids or your, you know, some kind of the things in your media, vitamins, etc. So they will sterile filter it. They will have a pore filter which will have this pore size of zero point 0.2 micrometer pore size. Then they will filter the media through the filter. So you will all your bacteria, all your contamination will be larger than 0.2 size. So it will not get filtered inside the fresh media. So here you can also see the storage temperature. It will be given in all the products that you get for the lab. You should always check that after you receive the product and immediately store it in that appropriate temperature. So here it is 2 to 8 degrees. Sometimes they may say like store in cool dry place, or store in room temperature, or store in ambient temperature. So depending upon the type of reagent or liquid or whatever it is, you should know where to store it. This is DMEM. This is usually for adherent cells like HEK, semi-adherent HEK, HeLa and all. So here also you can see their uh, components. Like here mainly they have said that this is plus D glucose, and then this is plus pyruvate. So they have components like this, and then you can see here they have this lot number, which batch it is, reference number. Like there are different type of DMEM again. There will be DMEM with high glucose, DMEM with normal glucose, DMEM with low glucose, zero glucose, DMEM plus or minus pyruvate, DMEM plus or minus glutamine. So depending on your experiment type, you might need a different one. There will also be media without phenol red also. If you don't want, if maybe phenol red is interfering in your experiment, you might want to exclude the phenol red. So for every type, every variation of the media, you will have a reference number. So it's always important to know the reference number of your product and order it based on the reference number. When you give it to a vendor, give the reference number, not the name, because with the same name, there might be 100 variations of the media. So always give the reference number. Okay, and then they have tagged it, certified it sterile, and then here also they have given you can store it between 4 to 8, that is your 4 degrees. Okay, 2 to 8, sorry. So you will store it in your 4 degrees. Liquid mammalian cell culture media is typically stored in 4 degrees in the dark to prevent microorganisms from growing and to prevent light from degrading the media. Before inoculating the media with cultures, you should warm it to the appropriate temperature for the cell growth. This point we also spoke in the previous session. So you should never ever give cold media to the cells. It will give temperature shock to your cells. So before adding the media, you have to warm it, pre-warm it, the entire bottle, or aliquate it in a small tube, and then warm the small tube in the water bath for at least 30 minutes to reach to 37 degrees. Check if the bottle is hot. You can easily understand, or check if the tube is hot, whether it has reached the 37 degrees, only then you should use it. Okay. Next, which of these is the most central organs of cardiac system? I think this question is there in your assignment.
Heart.
Heart, yes. So your cardiac system, what does it do? It pumps blood, it purifies your blood, it reoxygenates your blood, supplies fresh blood, collects uh, bad blood or carbon dioxide rich blood. So you see, your heart is the main organ that pumps and does all this. So your heart, from where does this oxygen carbon dioxide exchange happen? And then you, in your lungs, you get oxygen, and then you get your oxygenated blood, and that is pumped to all the organs. And from the same organs, the carbon dioxide is again carried back like this, and then your exchange happens in your lungs. So that is your cardiac system. Next up, which is used as pH in media. pH indicator in
Ma'am,
media.
phenol red.
Phenol red, yes. So I did not put proper animation, I guess. So anyways, in the previous session we discussed, and in the media, in the first question also we discussed what all components are there. There we talked about phenol red. So you can see how phenol red will change its color according to the pH. This is your optimum pH for growth. So here what they are trying to say is when you have to change your media and what you have to do with the tube which looks like these. So if it is in this color, you can leave it for a day. Okay, then after that next day again you have to check. So next day it will be like this. So you have to change the media now. You have to replenish and give it fresh media because this is all used up. So you have to give it fresh media in 1 or 2 days. And then if your media is this color, that means change or regas with CO2. Usually this color comes when you put it in the hood or when it is outside. So because it is not the appropriate amount of carbon dioxide, this will be the color of the media. But once you place it in the incubator, this color will change to this because your pH is now maintained by the 5% CO2. So if still after putting it in the incubator, if your media is still in this color, that means your oxygen with your carbon dioxide supply, there is some issue. You have to check it. Okay. But if your media changes to this bright yellow, that means there is contamination. You have to check it for contamination and change it immediately. It can be 1 or 2 days. Two ways. That is, it is contamination if it happens very quickly. But if you have forgotten to change media for like a week and then it is like this, then it may be due to, fully consumed, that is, and all the secreted things are there in that, your cells will be in stress. Okay, so how this phenol red works, you might have done the titration experiments if you had chemistry before. So it has the zwitterion configuration. Okay, so then it gets one anion and that exchange happens. So in zwitterion configuration, you know what is zwitterion, right? It will be neutral. So that time it will be yellow in color. But once it gets, the anionic configuration, then it will be in red color. So generally at neutral pH, this will be in anionic state. So after that, if the pH increases, what happens? Your proton levels will come down. It will become more alkaline. So your this phenol red will gain an anion to neutralize it. So that time when it gains another anion, it will be in dianion state. That time it will change to fuchsia pink or pink. So but then when again it goes to zwitterion, it will be yellow. Again if you neutralize it, again it will be red in color. Okay, this is how it works. So we are done with all the questions. If anybody wants a recap for any of the slide, or if you want to ask any questions, you can go ahead and ask me. I will be sharing this presentation with you, and if you have any further doubts or queries also, you can post it in the NPTEL page. You can ask there. Okay, if anyone has a question, you can ask me. Okay, if there are no questions, then we can end the session here. Okay, so
Thank you, ma'am.
yeah, you can leave, or if anyone still has a question, I'll wait for a minute. Other people can leave the class. Thank you, everyone.